Good morning, everyone. This is Dr. Harshavardhan Tehli, and I am working as an assistant professor, Department of Genetics and Plant Breeding, PGCA, RPCA, PUSA. So today we are going to discuss about the DNA extraction. So that how we are going to do the DNA extraction, its protocol, and some of the troubleshoots that we may encounter while doing the DNA extraction. So this is the flow chart that how we are going for DNA extraction. So you need to take one or two leaf sample, the very young leaf sample you need to take, and you need to grind using this pestilent mortar. You can see in the picture that these leaves are placed in this particular pestilent mortar, and you are going to grind it. Okay, so you need to put a few drops of CTAB and you need to grind it. Once you are doing the grinding, okay, so then you need to take this particular material, whatever the substance or liquid material is there, you need to take in 1.5 ml or 2 ml tube, a kind of tube. Okay, so one more thing here is if you are having the liquid nitrogen, then you may use the liquid nitrogen to crush the leaf sample. If you are not having the liquid nitrogen, then you may go for using the you may go for using this CTAP for grinding the leaf samples. Okay. So once you have grinded, you are going to take in this particular tube, plain tube, which is sterilized tubes. Okay. So all the material, liquid material, you are going to place in this particular tube. Once you are placing it, then you need to add the CTAB, which is of 800 microliter with a 2 microliter of beta mercaptoethanol. So you are going to add 800 microliter of this CTAB into this particular tube in the crushed leaf sample. So once you are doing this, then you need to put this particular tubes in the inside the trays. You can see here these are the trays which are available in order to place this particular tubes, and also you need to label the samples 1, 2, 3, 4 according to the number of samples that you are going for DNA extraction. So once you are placing those particular tubes inside this trays, so you need to incubate it. Okay, so this tray can be placed inside the hot water bath. So you need to ensure that this hot water bath is at 65 degrees Celsius. Okay, so before starting this grinding protocol and all, you need to make sure that this particular hot water bath is on, and it should reach 65 degrees Celsius. So once it reaches 65 degrees Celsius, then you need to place this sample, that is tray, inside this particular hot water bath for about 40 to 45 minutes. So intermittently what you need to do, you need to shake this particular samples. You need to shake this intermittently. So once the incubation is done, you need to remove the sample out. Then you need to add the CI, that means chloroform and isoamyl alcohol. So you are going to add this chloroform and isoamyl alcohol in the 24:1 ratio. So approximately of 800 microliter. So if you want to prepare for one particular sample, then you need to put the 765 something chloroform microliter and 32 microliter of isoamyl alcohol for one particular sample. So if you are preparing 10, 20, 30, 40, so accordingly you need to prepare this particular solution. So you are going to add the CI, that is chloroform and isoamyl alcohol. Once you are doing this, then you are going to shake using this shaker. That means you are going to place this particular samples inside the shaker. That means you are going to put on this particular shaker so that it will be getting shaked very well mixed for approximately 40 minutes. So once you are doing this, so then you need to go for centrifugation. That is, you need to put the sample, you need to take out this particular tubes and you need to place in this centrifuge. Okay, at 10,000 rpm for 12 minutes. Okay, so we are doing this particular shaking and centrifugation process for just a phase separation. That means you need to separate out that DNA part, that is aqueous part, and you need to separate that other particular matters like protein content, other phenolic compounds and all. So for that you need to go for centrifugation and at the rate of 10,000 rpm. So in order to get this phase separated. So you can see in the picture that your DNA part, that is aqueous phase, will be coming up, and the rest of the organic phases and phenolics will be settled at the bottom. So the upper part will be your the DNA of the sample. So what you need to do, you need to extract this upper part of the DNA, that means aqueous solution, that will be usually in white color. Okay, you need to suck it. So and you need to take it into a new tube. Okay, so of 2 ml tube. So once you are taking it, then you are going to add the isopropanol. You can see here. So you are going to add the isopropanol of 700 microliter. Okay, so once you are adding it, you need to just invert the tube up and down for a few seconds. So there will be the visible, so you can see the DNA threads at the end. So once you are adding this isopropanol, so why we are adding this isopropanol is, so the DNA will be like getting precipitated. The DNA threads will be formed here. So you can just see here that the DNA threads are visible. So once you are adding the isopropanol, you are going to see these DNA threads. And then again you are going to centrifuge it at the 10,000 rpm for the 5 minutes after adding up of isopropanol, so that the DNA will be precipitated. So before also, once we have gone for centrifugation in the previous slide, you can see here. So we have gone for centrifugation here at the 10,000 rpm. So it depends. So how much rpm you are going to keep, and how, like for how much minutes you are going for centrifugation, it depends upon what particular stage you are. If you are going for phase separation, that is aqueous phase separation and all, so you need to put more amount of centrifugation, and you need to like rpm, and you need to go for maximum amount of rpm. Okay, so if you are going for DNA precipitating, then I think for 5 minutes it is very much sufficient to centrifuge the samples, so that the DNA pellets will be formed. So you can see here the DNA pellets will be formed once you are going for centrifugation. Okay, so what you need to do, whatever the liquid amount will be there, that you need to discard. So supernatant, the liquid medium, you are going to discard it. So the DNA is settled at the bottom. You can see here, this is the DNA, so which is settled at the bottom. So once this is formed, what you need to do is, you need to go for ethanol washing, that is alcohol washing. So you need to use 150 microliter of like ethanol, and you need to put in this particular DNA pellet where the DNA pellet is formed, so that any other extra debris, plant materials are there, waste materials are there, that they will be getting wiped off. For that you need to do it for 2 to 3 times. So once you are doing it once you are going for ethanol washing for 2 to 3 times, so then your DNA pellet will be very much white and clean. So all the debris will be lost here. So once you are doing this, then you need to air dry those particular pellets. You can just see here, you need to open the lid, and then you need to place it for 1 hour, so that water or liquid content, everything will be dried off. So only thing is, your DNA will be settled at the bottom in the form of pellets. Okay, so once you are doing this, then you need to dissolve this DNA sample. So for that you need to add the TE buffer. So for around 100 microliter you can use. So in the DNA, the pellets, so you can put this 100 microliter in each tube, and you need to store it under the room temperature, so that these pellets what are there here, so it will be getting dissolved. So once the pellets are stored at the room temperature for 1 day or a whole night, okay, so the pellets will be dissolved here. So then what you need to do is, you need to go for RNase treatment. So in the next day morning when you are coming, so you need to do the RNase treatment. Why we are going for RNase treatment is, so your sample contains the DNA and also the RNA. So we no need to have that RNA in that particular tube. So that for to get rid of that RNA, so you need to go for RNase treatment. That means the RNase chemical is there, which you need to put it around 1.1 to 2 microliter, and then you need to incubate it for 30 minutes at 37 degrees Celsius, so that RNA degradation will be there. Okay, and one more thing here is the TE before that you are going to use, that usually consists of tris base and EDTA. That means you are going to take off 0.121 gram of tris base and 0.037 gram of EDTA in 100 ml, and you are going to adjust the pH at the 8. Okay, so this is how we are going to use this TE buffer, and how we are going to prepare this. So this is overall the DNA extraction procedure, how we are going to do and all. And this protocol actually depends upon what type of a crop it is, how the leaf tissue is there, whether it is soft or hard, how the phenolic compounds are there. Certain crops are there like cotton, where high amount of phenolic compounds are there. So they will be getting binded to DNA. So for that, what we need to do, we need to increase the concentration of a beta mercaptoethanol during the initial time. Okay, and if the sample are young but not mature, then you may go for a very less amount of beta mercaptoethanol here. Okay, and similarly this centrifugation, so it also depend upon the same thing, that is whether the crop is hard, the tissue is hard, or whether the tissue is soft. Okay, so in order to remove the debris and all, we need to go for a more amount of centrifugation, or you need to increase the RPM rate, or you need to increase the time, like that. It depends upon what type of a crop or what type of a leaf tissue it is. So this is all about the DNA extraction, how we are going to do. And the next thing is troubleshootings. So while you are going for DNA extraction, you might encounter several problems while extracting the DNA. So it starts from beginning itself during the tissue grinding. You can just see here. So when you are going for tissue grinding, so if your leaf sample is not crushed properly, then you might be getting up with the low quantity of the DNA. And similarly, like while adding the isopropanol, that means you are going to add about 700 to 800 microliter. So if you are adding it very inappropriate volume, then there might be no chances of that pellets will be visible. Okay, so that pellets will not be visible if you are not going for a good amount of isopropanol concentration. Similarly, so when you are going for alcohol washing, that is pellet washing, so you need to be very much careful so that there is no loss of that pellets will be there. And while adding any chemicals and mixing, you should see that no rigorous mixing is done. You need to do it gently. Okay, and one more thing is the if the crop is, if the leaf tissue is having high amount of polyphenol contents and all, then that particular DNA will not be white, it will be looking brown or yellow. So for that, as I said before, you need to use a more amount of beta-mercaptoethanol. And also RNase digestion. Is, if you are not going to use this RNase treatment, then the sample that you are going to have, that will be having the DNA as well as RNA. So when you will be going for running in the gel, then you may encounter like a different bands, intensities, and the problematic things and all there. Okay, and also the drying. So once you are keeping the sample for drying, okay, that is after this adding of a TE buffer, then you need to ensure that this particular sample is placed in the good condition. It's not that you are going to keep it in an oven or where the extreme temperatures are there, so in order to rush to dry the sample very early. So you should not do like that. And ultimately, if you are following the good protocol, and the DNA extraction will be good, but the thing is, again, there will be a problem in gel loading. Okay, so once you are preparing this once you are using this all protocol and you are extracting the DNA, but still while loading the sample in the gel, okay, if you are making any mistake in order to check the DNA concentration, that is quantification and all, so there might be chances of a loss of the DNA sample. So low DNA concentration will be yielded if you are not following the proper protocol of gel loading and all. So these are the some of the troubleshoots that we are going to encounter in DNA extraction process and all. Hope everything is fine. If still any doubts are there, then I will be available. You can also note down my number, that is 89710 You can easily reach out to me through WhatsApp. If you have any doubts, I will be available for the same. And in the next lecture, I will be discussing about the role of certain chemicals that are used in this DNA extraction process and all. So for as of this lecture, so I just wanted to discuss about DNA extraction procedure and what are the problems that we may encounter during the DNA extraction. So hope this is very much clear now. Thank you.